Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to talk about the second video of the translation in eukaryotes, and we call that initiation of translation. Okay, so in the previous video lecture, if you guys remember, we talked about the overview of the eukaryotic translation. In our view, we talked the introduction of translation and we talked the requirement for the translation. This is the second video lecture of this series, and in this video we are going to talk about the initiation of translation. So let me write eukaryotic initiation of translation. Okay, now in this video of eukaryotic initiation of translation, we are going to talk about the introduction of this eukaryotic initiation of translation. We are going to talk about the requirement that what we required in this stage, and we are also going to talk about the steps and which are involved in this case, and we are going to talk about the mechanism of eukaryotic initiation of translation. You know, it is very complicated and complex stage. So before starting this video lecture, you need to understand the prokaryotic initiation of translation. So if you missed this video lecture, it is present into my channel. You can go back and watch the prokaryotic initiation of translation. Once you get idea about that, then the eukaryotic initiation of translation will be very easy. First of all, let's start with the introduction. What is eukaryotic initiation of translation? Now, initiation of translation is the stage. As the name indicates, initiation means the start point. So initiation of translation means it is the start point of the protein synthesis. It is the stage in which the protein are synthesized. How? We have a messenger RNA. Inside the messenger RNA we have a codon in the form of 3 nucleotides. Each codon will code 1 amino acid, and then we have a transfer RNA, and we have the 2 subunit of the ribosomes. So when small subunit of the ribosomes having all the initiation complex with transfer RNA will attached to the 5' capping area of the eukaryotic mRNA, and then it's scanned to the start codon. And after that, the 40S subunit are bound here, then it will make the ATS complex. Now, due to this ATS complex, the protein synthesis will be started. So this is called eukaryotic initiation of translation. So how exactly it will occur, we will explain it later. Now, this is the introduction. Let's talk about the requirement, that what kind of things we required in this case. So let me write. If we talked about the requirements, so we need, let me write, mRNA having codon, we need tRNA having anticodon which are complementary to the mRNA nucleotide. Then we need ribosomes having 2 subunit. One is the large subunit, other is the small subunit. Large subunit is 60S while the small subunit is 40S. Then we required initiation factors, eukaryotic initiation factors. Okay, so these are the main things that we required in this initiation of translation. You know, if you guys remember, we talked in prokaryote, there are 3 initiation factors: IF1, IF2 and IF3. In eukaryotic initiation factor, we have almost 10 to 12 initiation factor. This E means, this E doesn't mean elongation, this E means eukaryotic. Okay, so eukaryotic initiation factor, we have almost 10 to 12 eukaryotic initiation factor, and the name are EIF1 and IF1A. Then we have EIF2 and 2B. Then we have EIF3. Then we have EIF4, 4A, B, E, F and G. And then we have EIF5, A and B. So these are actually the these are the eukaryotic initiation factor. Okay, so we have almost 10 to 12. So if we count this, so this is the first eukaryotic initiation factor 1 and eukaryotic initiation factor 1a. These are the two. Then we have eukaryotic initiation factor 2 and eukaryotic initiation factor 2b, so 4. Then we have eukaryotic initiation factor 3. So now if we add all these, we get 5. Then we have elongation initiation factor for A, B, E, F and G. So these actually make a complex. Now then we have eukaryotic initiation factor 5A and 5B. So if we count it, so let's count it. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So almost we have 12 initiation factor, eukaryotic initiation factor. Now let's talk about the function of each initiation factors. Now remember, initiation factor 1, 2 and 3. Sorry, initiation factor 1, 2b and 3. These are those initiation factor which are bind to the E site. Okay, then we have initiation factor A, 1a. Now initiation factor 1a, they will bind on the A site to prevent the immature transfer RNA to bind on the A site. Then we have initiation factor 2. This initiation factor 2 having the GTP like activity. So they will bring the charge transfer RNA onto the 40S subunit of the ribosome. Then we have initiation factor 4a. Now this initiation factor 4a having the RNA helicase like activity. Then we have initiation factor 4b. This initiation factor 4b having the activity, they will scan the mRNA and bind to the AUG site. It means that they will facilitate the scanning process. We will talk to it that how it will helping in scanning process. Then we have initiation factor 4e. So this initiation factor 4e will bind to the initiation this 5' capping of the mRNA. Then we have initiation factor 4g. It will the main function of this 4g, initiation factor 4g, they will bring the 3' polydenylation A site to the 5' capping area, due to which a loop is formed. And how the loop is actually important for the scanning, we will talk it later. Then we have initiation factor 5a. The initiation factor 5a is responsible to release or to dissociate all the other initiation factor on the mRNA. Then we have initiation factor 5b. This initiation factor 5b is GTPase like activity, and it will bring the large subunit to the what we can say to the small subunit. So large and small subunit are then connected with each other. So these are actually the function or a little bit function about the what we can say in is eukaryotic initiation factor. Then we have different steps that are involved in eukaryotic initiation of translation. So let me write each steps. Formation of 43S pre-initiation complex. So first of all, 43S pre-initiation complex will form. This is the first step. The second step is attachment of mRNA with this 43S pre-initiation complex. So let me write attachment of mRNA to 43S PIC, pre-initiation complex. Third, we have scanning of mRNA to the start codon. So scanning of mRNA to start codon like AUG. Fourth, we have formation of ATS complex. Formation of ATS complex. So these are the main steps that are involved in initiation of translation. Now let's talk the mechanism. First of all, the formation of 43S pre-initiation complex will form. How it will form, let's talk about that. You know what we have? We have this is the small subunit of the ribosomes. So we have small subunit of the ribosomes. What we have, so this will be the 40s. So we have 40s small subunit of the ribosome. Now in case of prokaryote, it will be 30s. Now what happened? In this case, we have also the 3 sites, as you can see. One is the E site, the mid one is the P site, while the last one is the A site. Now this is the A, P and E site. So with this E site, we have 3 initiation factor which is already binded here. But first of all, IF3 is already binded with this E site, as you can see. So this is, let's suppose, IF3 binded here. This green dot can represent IF3. Then we have some other initiation factor like IF2B and IF2B and IF21. So let me write. So what we have 3 initiation factor like IF1, 2A and 3, 2B and 3. So these 3 initiation factor are binded to the E site, but among them the 3 will already binded here, and after that the initiation factor 1 and initiation factor 2B will bind to the E site. Why it bind to the E site? Because it will prevent the large subunit, which we call that 60s subunit, to bind on the 40s site. So it will prevent that phenomena. Now then we have the A site. So on the A site we have already another initiation factor bind, which we call that IF1A. IF1A. So initiation factor 1A will bind to the A site. It will prevent the immature charge transfer RNA to bind on the A site. Okay, so this is the situation of the 40s. This is the 40s subunit of the ribosome, and all these initiation factor are binded here. So it will get activated. Okay, you know what we have? We have already the ribosomes are present inside the cell. So inside the cell, the ribosomes is already present. Then after that, what happened? The next initiation factor will get activated. So after that, a next initiation factor will get activated, and we call it initiation factor 2. Initiation factor 2 will have GTPase like activity. So let me draw here. So this is the initiation factor 2. So what they have GTPase like activity. Okay, so let me draw here. They have GTP. Okay, with this IF2 they have also the transfer RNA, as you can see. So they will carry the charge transfer RNA. Okay, now this transfer RNA is already charged. It means that amino cell transfer RNA synthetase will bring the amino acid and it will bind it to the 3' area of the transfer RNA, due to which the transfer RNA are become charged. Now after that, IF2 will bind to this charge transfer RNA and it will carry this charge transfer RNA. Now it will carry this charge transfer RNA to this 40s subunit. And after that, it will bind to this 40s subunit. So what we get the situation like that. Okay, so we will get the structure like that. This is the GTP. Okay, so now let me explain. This is called as 43S pre-initiation complex. PIC means pre-initiation complex. How it will form, let me once again tell you. So this is the first stage, formation of 43S pre-initiation complex. First of all, we have this 40S subunit of the ribosome, which have the initiation factor like IF1, 2B and IF3, which bind to the E site, that prevent the large subunit to bind here. Then we have A site, which is blocked by IF1A. So due to this initiation factor, it will prevent the uncharged transfer RNA or immature transfer RNA to bind at that specific area. And then we have P side, which is already freed. It will get activated. This 40S subunit once get activated, then after that a next initiation factor 2 will get activated. Now once initiation factor 2 having the GTP, it will bring this charge transfer RNA, as you can see, and it will bring this charge transfer RNA to bind with this 40S subunit, and finally we get 43S pre-initiation complex. So this is the formation of 43 pre-initiation complex. Then next what happened? Next we have the attachment of mRNA to 43S pre-initiation complex. So let me draw the mRNA here. So this is, let's suppose, the mRNA. This is the 5' area, so all of you better know the 5' area is capped. So the 5' area is methylated capped. And this is, let's suppose, the start area, which we called it AUG site. And after that we have the stop codon UUA, and after that we have the adenine polyadenylation. So this is the 5' area and this is the 3' area. Now this is the mRNA, eukaryotic mRNA. What happened in this case? This is, let's suppose, the eukaryotic mRNA. So after that what happened? The next initiation factor will be added here. So initiation factor 4 complex will bind it here. It means that initiation factor 4 A, B, E, F, G will recruit it here. So let me write A, B, E, F, G. So all these 4 initiation factor will recruit it here, and what we get the structure like that. So let me draw. Okay, so this is the first initiation factor, which we call that initiation factor 4E. So first initiation factor 4E will bind to the 5' capping area of this eukaryotic mRNA. After that what we have 4A. 4A will bind to this specific area of 4A will just bind to this IF4E site. Then we have 4G. So 4G will bind to this specific area, 4G. Then we have 4B. So 4B will just bind to this site, 4B. So these are the complex, as you can see. Okay, so what we have initiation factor 4E which bind to the 5' capping area. Then we have initiation factor 4A which bind to this initiation factor 4E, and then we have initiation factor 4G which bind to this A, and after that we have initiation factor 4B which bind to this start codon area. Now then what happened after that? These are the complex which are binded to these sites. After that what happened? This 4G, as you can see, this 4G will bring this polyadenylation to this 5' area, and what we get a loop like structure. Okay, so the situation will be like that. So it will bring closer to this polyadenylation to this 5' area, as you can see. Now after that what we have, so let me draw each initiation factors. This is the initiation factor 4E, this is the 4A, this is the 4B, and this is the 4G. Okay, now these are the initiation factors, these which are binded here. After that what happened? This 43S pre-initiation complex, now once this situation will formed, as you can see, once the loop is formed, then this complex, means this RNA will attach to this pre-initiation complex, 43S pre-initiation complex, and we will get the structure like that. Okay, so then we will get the situation like that. Now let me explain that what happened. Once the loop is formed, so then this loop like structure of the RNA will just attach to this 43S pre-initiation complex of this subunit of the ribosome. Once the attachment is possible, so we will get the structure like that. So this is the 43S initiation complex. As you can see, they have all these initiation factor like initiation factor 1, 2b and 3, and they have initiation factor 1a, and moreover they have initiation factor 2 which carry the charge transfer RNA, as you can see, and they have also the GTP. Now it will just bind here. As you can see, the transfer RNA is still not properly binded, so it will be look like in this form. After that, what happened, the scanning will occur. So this is the attachment. Now the attachment is done. The next process is the scanning. So the next step is the scanning. After that, the scanning will occur. So after that, this whole complex, as you can see, we call that 48S initiation complex. So we get 48S initiation complex. What happened in this case? During scanning of mRNA to AUG site, this G and B, okay, remember this G and B means initiation factor for G and initiation factor for B. They will help in the scanning. They will, proceed or move this 43S initiation complex to the AUG site. So we can say that for B and G are just help to scan or just help to move this 43S pre-initiation complex to the AUG site. So it will facilitate the scanning process. Now after that, once it reaches to the AUG site, so then a proper attachment of this transfer RNA to the start codon will occur. So after that, what we get the situation like that. So let's suppose this is the 4E initiation factor 4E having also this initiation factor 4A, and moreover these are the complexes which moved. So after now this is the start codon, as you can see, A U U G, and this is the stop codon UA A. These are the polyadenylation area. So let's suppose this is the complexes. Now this is the small subunit of the ribosome. As you can see, it will just come here. So let's suppose this is the start codon AUG. Okay, now this is the P site, A site and E site, and they have also the initiation factors, as you can see. Now remember guys, when the 43S, let me draw the other initiation factor. These are the initiation factor for G, and let's suppose this is the initiation factor for B and F, let's suppose. Then what happened, once the situation will like that, once this small subunit of the ribosome reach, and during scanning, once it reach to this start codon, as you can see, to this AUG site, then what happen, the transfer RNA, which is previously like in this shape, it will be correct binded or properly bind to this AUG site, to this start codon. Now it will be correctly bound here. After that, what happen, the GTP, as you can see, it will be converted into and hydrolyzes into GTP plus inorganic phosphate. So then GTP will be released out. Okay, so this is the situation like that. So this is called a scanning of mRNA to start codon area. Now it will scan or reach to the start codon. After that, the last step will occur, which we call it formation of ATS complex. Then what happen? The large subunit, which we call it 60S, will get activated, and it will be brought by this initiation factor 5A. So it will be brought, and we can say that initiation factor 5B. The large subunit was brought by this initiation factor 5B. So we can say this initiation factor 5B acting as a GTPase. Okay. Now what happened? We have, let me draw here. So let's suppose this is the large subunit. As you can see, they have also this GTP and what we call that IFB. So before that, what happened? Before that, the initiation factor 5A. So this initiation factor 5A will get activated, and it will just bind here to it is very huge complex or protein. It will just bind here, and once it bind here, so then what actually it will going to do, it will release all the initiation factors, like initiation factor 1, initiation factor 2, initiation factor 3, initiation factor 4, initiation factor 1a, initiation factor 2a, initiation factor 3, initiation factor 4a, b, c, d, e, f, g. So all the initiation factors will be released out with the help of this initiation factor 5a. Once all their initiation factors are released out, so now this large subunit will be binded here, and we will get the structure like that. So after that, what happened, the loop will also be dissociated, or the loop will also be, disappear. So this is, let's suppose, the capping area. This is, let's suppose, the start codon. These are the stop codon, and these are the polyadenylation area. So what we get the structure like that. These are the, again, the small subunit. Now this large subunit will be brought by this IF, initiation factor 5b, and once it bind here, so then what happen, the GTP will be converted into GDP. After that, again, inorganic phosphate will also formed, or pyrophosphate, in which the energy will be used. And after that, what happened, let's suppose these are the site, as you can see, the site 1, this is the site 2, and this is the site 3. So this is the E site, A site, and P site. Sorry, A site and P site. Okay, and here we have the transfer RNA which carry the amino acid, F-methionine amino acid. Now this is the situation, and this is called as formation of ATS complex. So after that, the 40S and ATS will be formed. So what we left it the complex, ATS complex. So in ATS complex, what we have the mRNA, we have the 2 subunit of the ribosome, we have the what we can say, this charged transfer RNA, and A side is get freed, E side is get freed. So all the initiation factor which is bind to E side and A side will be released out. So A side and E side are now freed. So new transfer RNA, charge transfer RNA, will bind here, and thus the elongation of translation will be started. So this is all about the eukaryotic initiation of translation. I hope you guys get idea about the eukaryotic initiation of translation, but if still you have any kind of question in your mind, you can write it into a comment. I will give you answer as soon as possible. If you like this video, so make sure to hit the like button, share this video, and definitely subscribe the channel, and stay tuned, because in the next video lecture we are going to talk about eukaryotic elongation of translation in a detail. So please keep watching. Thank you so much.